In order to build a clock, a common strategy is to start with an oscillator that has a regular period, a beat. Now, perfect negative feedback gives you a set point control. Think of how hormones, glucagon and insulin, maintain your blood sugar at a steady level even though you only eat 3 times a day. But negative feedback with an intrinsic delay turns out to give you an oscillation around a set point. Sometime around 1980, this fellow was teaching students at the Woods Hole Marine Biological Laboratory how to run protein gels, extract proteins from cells and run out the soup on a gel to separate them into individual molecular species. And they were working with sea urchin eggs and clam eggs and taking timed extracts as the embryos underwent cell division. And they observed that the levels of a particular protein went something like this. They would climb as cells approached division, then abruptly decline as cells went through division. Where this point here approximately corresponds to the metaphase-anaphase transition, that is, when chromosomes start to segregate along the mitotic apparatus. MPF activity, in turn, during the cell cycle, has a somewhat different profile. It's not there at all, and then abruptly, MPF activity develops in the cell before collapsing as the cells go through division. Remember, MPF is this activity that you can translate from one, say, maturing starfish oocyte to another immature starfish oocyte and induce maturation. So the green thing here, that's MPF activity, rising and falling in this sort of abrupt sawtooth sort of way. And this here is a protein that came to be called cyclin, for obvious reasons. MPF, recall the M originally stood for maturation and it came to stand for mitosis. MPF turned out to be a complex between cyclin B and a protein kinase called cyclin-dependent kinase, number 1. So let's draw out now how these work together. I'm going to draw them as two sort of jigsaw puzzle pieces in a way. That together make one interesting species. So this happens to be the cyclin half of this creature, and this is the CDK half, the business end, as it were. Before we get too much further, this protein is phosphorylated by a kinase that inhibits its activity. So there's a clamp on its jaws. That has to be removed before this MPF is active. Okay, it's not enough to just get cyclin and CDK together. those inhibitory phosphates are removed by a phosphatase called CDC25 to yield the active enzyme. The active MPF has a number of targets in the cell, but the first one that's relevant to us is CDC25. MPF activity activates CDC25, which activates more MPF activity. Okay, so that's a positive feedback loop. More of this makes more of this, which makes more of this, which makes more of this, etc. That's the basis for the effect in which transplanting some cytoplasm with a little MPF into it from a maturing starfish egg into an immature starfish oocyte can prompt activation of latent MPF within the cytoplasm of the recipient, and so on and so forth. But let's see first, before we go around that way, where these phosphates came from. This complex was created, of course, by cyclin joining with CDK to form the inhibited cyclin CDK complex. These phosphates were added by a kinase called WEE1. WEE1 was discovered in a mutant screen in yeast, and because this WEE1 protein is missing, the cells enter mitosis too soon, and you get wee little yeasties, okay? I'll deal that, so slightly better. Okay, so WEE1 inhibits the enzymatic activity, of CDK. Where did the cyclin bit go? How did it lose its tail? As the cyclin CDK complex goes around in cells finding its targets and phosphorylating them in order to drive the cell cycle through M phase, it acquires a tag. The tag is applied to the cyclin portion in the form of a chain of ubiquitins. Ubiquitin is a small protein that is, you guessed it from the name, ubiquitous. And the ubiquitin tag serves to mark proteins for degradation. The ubiquitin tagged cyclin B is destroyed by the cell's general protease garbage collector, the proteasome. The proteasome is this big barrel, and proteins are sort of fed in at one end and chopped up into bits. Ubiquitin doesn't just attach to any old protein. Ubiquitin is added by a so-called ubiquitin ligase enzyme, an enzyme that links ubiquitin to specific sites to target proteins for degradation. And the one that promotes ubiquitination of cyclin is called the APC, for anaphase promoting complex. That is a ubiquitin ligase. So the active MPF has a variety of targets during M phase, histones that promote chromosome condensation, lamins in the nucleus that there's phosphorylation prompts nuclear envelope breakdown, all kinds of microtubule binding proteins. Very many things are targeted by MPF to change the physiology of the cell. One of its targets is this APC. Now, should that be a positive or a negative? Let's see, MPF is targeted for destruction by the APC. It turns out this is a positive. MPF indirectly stimulates its own inactivation. Now we need one more link here to complete the picture, roughly. And that's from MPF to WEE1. MPF inhibits the activity of WEE1. That is, it inhibits its own inhibitor in this case. This life cycle here of the MPF, of the cyclin CDK complex, consists of a series of intertwined feedback loops. One of these is a positive feedback loop between CDC25 and CDK. They activate each other. In contrast, WEE1, by phosphorylating and preventing the enzymatic activity of CDK1, inhibits MPF, but MPF in turn inhibits WEE1. Okay, so we have a double negative there. You can sort of see where this is going. If one of these is active, they're both going to be active, right? If they get across whatever thresholds exist. In contrast, only one of these is likely to be active at once, right? The third feedback loop here is a straightforward negative feedback loop between APC and CDK. And again, CDK activity promotes the activity of the anaphase promoting complex, which via ubiquitination inhibits the activity of CDK1 by stimulating the destruction of the essential subunit. I want to provide a short sketch of a dynamical systems view of the cell cycle clock that I just sketched out as sharks and arrows, using differential equations. Don't be afraid. The differential equations are very straightforward. They all say there's some rate that's a function of some parameters and concentrations of molecular species in the cell, variables of whatever kind. So we're going to have the rate of change of some concentration some molecular species, proteins or whatever, proteins in this case, for any molecular species in the cell, whether it's involved in the cell cycle or not. It's going to equal some balance of synthesis rate, right? And a decay rate. You're going to add to the concentration by how fast it's synthesized and take away from it by how fast it's degraded or chopped up by, some specific targeting. And there might be other additive terms in here, other processes. For example, that molecular species might be converted to another form, or might be produced from another form, or something like that. Okay, so the symbolic version of this is to say, the rate of change of the concentration of A could be ATP could, or the A could stand for aspartate transcarbamylase or whatever. In this case, it's just going to be A for now. And let's say this is a gene product, and we'll forget about the difference between transcription and translation and pretend, just for the sake of simplifying this, that we can make a gene product in one step. How is it synthesized? Well, it's transcribed. There has to be some maximum rate of transcription, right? Some maximum rate that RNA polymerase can go. And then there has to be some efficiency that's determined by gene regulators, right? Whether that particular gene is turned on by some sort of transcription factor or not. And I'm just going to say, instead of trying to write something out, I'm going to say it's going to look something like this. This is going to be a function, and it's going to have this general shape, where this plateau is 100%, otherwise known as 1. This is a very general rule about biology, okay? These kinds of dose response curves, they don't just go up and up and up forever, right? There's some sort of maximal rate that this process can happen, and the effect of regulatory proteins or even nutritional state or something like this is to determine how efficiently you achieve that maximal rate. So the rest of the equation, minus a decay rate. And if nothing else is going on, every molecular species in the cell has a half-life. It might be long. The half-life of some proteins is probably measured in days. Some proteins, the half-life is measured in minutes. But at any rate, there's some degradation term there. Okay, and let's just say that this so-called sigmoid curve is a term that looks like this. This is really convenient. Let's say it's a function of some regulator, B. There's going to be a constant in there. This should be familiar to all of you who've taken introductory biology, because this is the kind of formula that would have applied to the binding of oxygen by hemoglobin, or the regulation of the lac operon by the lac repressor, or something like that, as long as we have a little exponent in here. If it weren't for that exponent, or rather, if that exponent, this little Greek letter that I stuck in there, this curve would just be a linear curve that eventually keeled over to a plateau. That's if this exponent is 1. But if it's a number bigger than 1, we're going to get a curve, a sigmoid shape like this. The exponent is due to what we call cooperative binding, and it's a very widespread aspect of allosteric regulation in nearly all of the transactions of macromolecules. Okay, so let's say we've got B to think about as well. And we'll just talk about a simple positive feedback loop first. Let's say that B is produced at a rate that depends on A. So we're thinking about A, which promotes the production of B, which in turn promotes the production of more A. So the maximum rate is going to be multiplied again by one of these. And the exact proportions might differ for B, of course, than for A. The twist of this curve might be different. B is going to have its own half-life. None of that makes any qualitative difference really at the moment for this, for the point of this argument. Here's how we think about this provisionally. Steady state, that is, when things aren't changing, when the concentration of A and B is steady, that's when this term here is equal to this term here, right? That is when degradation balances production. I.e., when equals 0, then we must, of course, have that if I adopt this particular formula, we must have this steady state condition. Remember, K here is just a constant, and its physical meaning is this. K is the concentration of B at which you get half maximal response. So K just says, how much B does it take to get halfway up this curve? We can write down the same thing for B. It's completely symmetric in this case. Now I'm going to just do one little rearrangement here, because obviously I can multiply the right side by the denominator over here to say that at steady state, the concentration of A is its half-life times its synthesis rate. And that makes a good deal of sense, right? If the half-life is a large number, that is, if the half-life is long, it takes a long time for this product to turn over, then A should be able to accumulate to a higher level than if the half-life is short. If A gets chewed up almost as soon as it's made, then the steady state level of A is going to be very low, right? At any rate, this is a steady state concentration of A as a function of B and some constants. So we can plot that out, right? And of course, the curve is shaped like this. We can have the concentration of A here and the concentration of B down here. And since we have the same for B, we can also plot B as a function of A at steady state. We're going to get two curves. They're going to look like this. That's just this curve for A as a function of B, and the same curve reflected for B as a concentration of A at steady state. Okay, these cross. These curves have a name. If you happen to go to parties with mathematicians, you can throw around this new word if mathematicians go to parties. These are the nullquines. And where the nullquines cross, both dA dt and dB dt equals 0. Because remember, we got these curves by saying, well, okay, what happens when this is equal to 0 and what happens when this is equal to 0, right? So wherever these null lines cross, both A and B are at steady state. Some further analysis that I won't trouble you with will show that this steady state down here, where they're both off, is stable, meaning that if you start out with this system somewhere down here, and you give it a little nudge over here, it will go right back to this steady state where there's none of A or B present. This one is also stable. This steady state is unstable, meaning if you happened to sit right there, right at the crossover point, nothing would be changing but a little tiny nudge in any direction away from that steady state point, and the system would zoom off this way, turning off in effect, or zoom off this way, turning on. What if we have A and B inhibiting each other? We have to have a little bit of an extra thing here. We have to say that there's something else that by default we're producing each of these, let's say, so that maybe we have a little trickle of each of them, but if you get a little more A, it'll squelch B, or if you get a little more B, it'll squelch A, and so on. Intuitively, we already said this ought to make an either-or switch. Either you ought to be able to achieve a state where the A is high and the B is low, or the B is low and the A is high. Well, what's the arithmetic going to look like? Instead of a term like this, if B inhibits production of A, then you'll have something like 1 minus. This means that term becomes 1 minus the same thing. Perfect. What's that going to look like? Well, 1 minus this curve just means flipping it over like that, okay? So that means instead of a sigmoid ramp climbing up from 0 and plateauing at 100%, if this is our 100% here, we'll start out at 100%. And the more B you add, the less production of A you get out. So I'm going to draw the null lines for this situation down here. I'm going to make a row of 3 of them across the bottom. Now, I should admit that it does depend on the careful choice of the values for these parameters. They could be way off so that none of this makes any sense, right? But give me the benefit of the doubt here. Again, we get 3 points where the null lines cross. That is 3 steady state points where neither A nor B are changing their concentration. You either have A high and B low, or B high and A low, right? And you already can imagine this is what we call a saddle point. This is an unstable steady state point. You could sit at the top of the mountain pass indefinitely unless someone gives you a nudge off of it, and then you go one way or the other. Either you roll down to the western side of the mountain range or to the eastern side of the mountain range, right? These are stable steady state points. we have made a rationalization for an either-or switch based on two molecular species that inhibit each other's production. Finally, this is our third case, of course. If A promotes the production of B, but B inhibits the production of A, then for the effect of B on A, we're going to have a function that looks like this. And for the effect of A on B, we're going to have a function that looks like this, right? So B as a function of A looks like that. A as a function of B looks like that. These milk lines cross in only one place. That is a stable, steady state point. It is an intermediate concentration of both of them. As I said, perfect negative feedback would give you a set point. Now, the little trick that makes an oscillator is that there are always delays. The furnace in your house isn't perfect either. There's a little delay in the thermostat. There's some delay due to drafts or the positions of the ducts or because someone parked a sofa in front of one of the heat vents or something like that. That's why the temperature in your house goes up and down instead of remaining at a perfect 68 degrees Fahrenheit or something like that, whatever happens to make you comfortable. Okay, so the effect of delays in this circuit is to produce a little bit of an overshoot. Instead of just zooming in directly to that steady state point from wherever you start, what really happens in a circuit that involves delays is that there's what we call a limit cycle. Trajectories wind in and then they orbit. They orbit that steady state point indefinitely. So these represent the three basic units that I said the cell cycle oscillator is made from. This represents the relationship between CDK1 and the APC, except I wrote it backwards, but we'll forgive that, won't we? This represents the basic relationship between CDK1 and WEE1. And of course, this is the basic relationship between CDC25 and CDK. Sorry for the crowding there. What effect does that have in the context of these diagrams? Well, perhaps you can imagine if we start out with an oscillation based on some negative feedback loop, if we start out with an oscillation, what we really want is something that's quite definitive here. We want a larger amplitude to that oscillation. We want it to be more definite when MPF is active and when there's just none of that activity around, okay? So you can imagine that entangling it with a switch-like behavior like this might pull that limit cycle out, distend it in this way. And you might further imagine that if you entangle it with an either or type circuit like that, you could have to choose another color. You could stretch it further. So that the limit cycle bloomed out into a great big excursion through this so-called state space. And that is basically why the life cycle of MPF is rigged up the way it is. The entwinement of these feedback loops, these represent dynamical modules that confer this kind of large amplitude on-off switch-like pulse behavior to the activity of MPF.